Hello, everyone. I can slowly start to see people joining. So we will wait about a minute until everybody manages to transition from the waiting room to the main room of our webinar. But we are very excited for our guest today, Dr. Mahdi Moqri. People are still joining. Oh, wow. People were really excited for you, Mahdi. That's the one of the highest numbers we've ever got so far.
Thank you.
It's beautiful. Numbers skyrocketing and now leveling. Okay, so that's the official time to start. Hello, everybody. My name is Dominika Wilczok. I am the coordinator of the Longevity Education Hub, and I am very happy to welcome you at today's webinar hosted by Dr. Evelyne Bischof, featuring Dr. Mahdi Moqri. We will be talking about validating the clinical biomarkers of aging. And with that, let me give the floor over to Dr. Evelyne Bischof.
Thank you so much, Dominika. Dominika is our leading force in Longevity Education Hub and excellent student at Duke and longevity author. But I'm so grateful and honored to introduce Dr. Mahdi Moqri from Harvard. He's at the lab of Vladimir Gadyshev, Professor Gadyshev, and also at the Snyder's lab in Stanford. He's a part of the medical faculty of Brigham's and Women's and is also the director and leader of the Biomarkers of Aging Consortium, which we will hear about more today, and which I'm sure every each of you knows. That's why you are here, or will be listening later. I would just like to add, personally, that Dr. Moqri is also very passionate mentor and educator. And so it goes very much with the Longevity Education Hub. Education is something usually very under the table, and people don't appreciate it publicly enough. So I would like to do that here, and I think it's so crucial and important to create, a good program for a new generation in aging research and aging science and longevity medicine ultimately. But I will now turn over to Dr. Moqri directly. Once again, thank you so much for your time and your busy schedule to be with us and to give us this piece of knowledge from yourself. Thank you.
Thank you very much, Evelyne. Thank you, Dominika. Thank you for inviting me and the opportunity. I'll try to be brief, going over some of the updates that we have with research and with the consortium, maybe for 25 minutes and just or less. Let's see how fast I can talk, and then maybe some minutes for questions. So let me just see if I can share my slides. Okay, do you see my screen in the full screen? Perfect.
Yes, everything works perfect.
Great. So I'm a junior faculty at Harvard, affiliated with Broad, an investigator with the Brigham Hospital, and still having some of my time with the Stanford. And I'm also a co-director of the Biomarkers of Aging Consortium, which I'll talk more about. And I'm pretty much academic. I've been spending some time. My training was in computer science and data science, and my faculty position, first one was in informatics, and then I fell in love with biology and aging, and I did the first postdoc with Mike Snyder group and Vittorio Sebastiano at Stanford, and for the past 3 years, I've been a research staff and more recently a faculty at Harvard Medical. And maybe my first encounter with aging in a research setting was during my first graduate thesis in Boston. When we worked at the hospital, we looked at all the medical records from several thousand patients who got admitted, who got entered the emergency department, and the hospital wanted to know how they can use AI or machine learning to reduce their ED boarding, where it's a known issue with most of the emergency department, but a lot of patients come for a visit, but only 30% gets admitted and they go to operating room or ICU. And we looked at different factors like encounter reason, and you can see different encounter reasons have different admission rates. But most interestingly, when we put everything together, including encounter reasons and demographics and everything, the top predictive factor for patient outcome by far was the age of the patient. And that was very interesting, but it came back to me when I started my research group as a faculty and building more advanced models for ICUs across the country with large number of patients data from large data aggregators. And we looked at different domains like accidents or cardiovasculars or neurologic. And we looked at all the data now, including text analysis and everything on the medical records and beyond. And again, interestingly, age was always among the top factors in various domains. And this happened in other research when we built models for cancers or Alzheimer's, not just in patients, but overall. So much so that I decided to take some time off. I started learning about the biology of aging very early on during my tenure track. And I came across this beautiful set of research at Stanford and Harvard and in Europe that they showed there are some cellular interventions, especially cellular epigenetic reprogramming, that reduces the epigenetic age of the cells. Here on the y-axis, we have DNA methylation age. And it was an interesting concept to me as an informatician. I was excited to see that machine learning informatics measure is being used to capture the age or biological age of cells and tissues and organs. And at the same time, that there is an intervention, at least one intervention that seems to consistently and reliably reduce this composite measure. So I took a sabbatical. I joined the Stanford Sebastiano groups to continue working and extending these kind of measures, epigenetic age as a whole. And I used some machine learning method to model the epigenetic landscape of cells based on all the features that we can extract, including genomic and other features and the age. And the idea is that some specific regions of the genome are more influenced as we age or as the cells age or even as the cells replicate. And we found that specific patterns, in particular binding of PRC2 complex, which is a repressor, coincide by the change in the epigenetic landscape by age. And here I'm showing one example that looks at skin cells. And if you look at the young samples, skin from young donors, the epigenome is on average low in these regions. But if you look at the old samples, the epigenome, this composite measure, is much higher. And you can also see that the sun-exposed regions, the sun-exposed skin samples are even higher than the sun-protected. So at the same time, capturing age and age-dependent exposure. And we validated this across many cohorts and cell types in human, in mouse, even in chicken. So it seems to be conserved mechanism, biological mechanism. And we end up using this in the lab and beyond to get more insight about these cellular interventions. Here on the left, you can see a modified version of epigenetic reprogramming, and we end up using our more advanced epigenetic methods to see which intervention is more likely to rejuvenate the cells. But this was an example of biologically interpretable and meaningful biomarker of aging, but turned out to be less useful clinically in the sense of longevity clinics or clinical trials, because the association with all-cause mortality or various age-related outcome was not as high as we hoped for. And at the same time, when I presented this around 3 years ago at Cold Spring Harbor and some other conferences, I also realized there are a lot of activities in the longevity clinical space using similar epigenetic clocks or other biomarkers as a measure that is expected to guide the intervention. So this is a secondary outcome that if positive or if reduced, the clinical trial is expected to be beneficial. But at the time, it was very concerning to me and some other folks who were developing these methods, because these models are not designed or trained to capture an intervention, or we hadn't proved that reducing these measures would be beneficial in a clinical trial. And if you can see here, they've been used for lifestyle intervention, pharmaceutical intervention, supplementary, and this was a few years back, and now it's much more common. And at the same time, in the space of aging biology, we know there's a lot of disagreement and on the fundamental concepts. We still don't know what is biological age, what is a good definition, or what could be classified as a biomarker of aging. In other fields, in cancer, in other diseases, we have very well-defined biomarkers. We have metrics to evaluate these biomarkers, but in the field of aging, it's still early on. And what we did is we invited the diverse team of experts who are involved in either developing or validating or implementing these biomarkers from various groups globally. And we invited everyone to work together to develop some consensus around these biomarkers and how we can validate them and how we can reliably use them in clinics down the line. And the first consensus work was about application, classification, definitions, very basic consensus that took us actually the longest. It took 6 months just to come up with a good definition of a biomarker of aging. But the main message, the main result, the main outcome of that work was now we have this classification of biomarkers that to some extent are adopted from FDA framework. And now we know what are the application context of use, and we have better language to describe these biomarkers. Are we talking about predictive powers? Are we talking about response? And in particular, if you look at the response biomarkers, we need this intervention to be, we need the biomarker to be responsive to an intervention. And if you want to use response biomarkers in a clinical trial, we need to have this biomarker in a causal pathway from the intervention to health outcome, which needs to be proved. And in the context of predictive biomarkers, we still need to validate these predictive associations across diverse cohorts, and we need to perform head-to-head comparison of various biomarkers similar to other fields to establish the predictive performance of these biomarkers. So we propose a framework to report these metrics in a more standardized way. And we also hosted these larger events, conferences and symposium. And last event was at Harvard Medical in November. And this year we are hosting another conference in October at Harvard Medical, where people come and present their latest research in the field. And after every meeting, we also report the survey of the field, and what people think and what people believe are the promises and challenges and the next steps and the roadmap to clinical translation of these biomarkers. And all of these are now public for everyone to read. And beyond the conceptual work, here I'm also getting into some of the work that we do in the lab, but first I start with this competition that we hosted with the consortium. Beyond the conceptual framework and consensus building, we also support these benchmarking efforts. And for that, we need data. One of the data sets that large data sets that we have access to is the data from Mass General Brigham Biobank. And the data set itself, the medical records, contains information from 15 million patients from 13 hospitals affiliated with Harvard Medical going back to 1850. And these are very well curated, all of the electronic health records, all of the genomic data, sequencing, and surveys, and all of the outcomes. The Department of Data Science and Informatics has spent around 20 years to curate all these phenotypes because ICD codes are many times not accurate, especially for some conditions. More than half of the time, the ICD alone is not accurate. So it takes a lot of effort to go through medications and notes and everything to have the correct labels for these conditions. Now, past 10-15 years, around 1,500 participants also donated samples, and a lot of them are just healthy donors coming for a checkup, medical checkup. And now we have a biobank of DNA, plasma, cells, and most of these participants are sequenced, either whole genome or genotype. And from this biobank, we picked 1,000 blood samples, and we profiled them in various ways, generating deep omics, high quality omics data, including methylation, proteomics, metabolomics, with support from some of the assaying companies and some foundations. And I want to also acknowledge Dr. Epigenetic, who is the co-director of the consortium, who was instrumental in this work. And we also raised some prize for a competition to host a data science, open science competition for the community to use these molecular data to see if they can predict the age-related outcomes. And the first round of the competition was just simply use molecular data to predict the age of the donors. And the second round was using molecular data to predict future survival, up to 10 years survival outcome. And more than 20% of these participants had passed away at the time of publishing the molecular data. It was a diverse set of participants from different backgrounds, mostly representing the New England, Boston area. And here I'm just showing the top 5 prediction results using proteomics or DNA methylation. And these are scores based on area under the curve, almost similar to percentage accuracy. And you can see that both proteomics and methylation data can now predict almost with 80% accuracy future survival, 10-year survival of the participants in a blind way. No one had access to the outcome. So even on the training, it was in a blind way. And you can learn more about these competitions and the open source library on the consortium website. But beyond these open science effort. These data and other biobank data are also being used to develop new machine learning composite scores for predicting outcomes, predicting age-related outcomes and diseases of aging based on these molecular data. And here you can see the same biobank data, MGB biobank that's used by Dr. Lasky's Su group at Harvard and in collaboration with True Diagnostic, and Dr. Gladyshev and I also contributed to this work. And on the left bottom, you can see how strong are these associations. If you create scores based on medical records and you stratify patients based on their scores, you can see the top scoring group are very well surviving after 10 years and more, but the lowest score group are dying more than half, more than 60%. So survival rate is now below 40% for a bottom group. And then these medical record-based scores could be also proxied based on DNA methylation, for example, on the right side, on the middle graph. And you can see high accuracy, moderate to high accuracy for predicting all-cause mortality using just DNA methylation. And you can see improvement over existing models, Grimmage being one of the top models. And here it's a proof of concept that higher quality and larger number of data sets could improve these predictive biomarkers, potentially could be used in monitoring healthy longevity. And on the right, you can see that these scores predict not just all-cause mortality due to one disease, but it seems to be capturing diverse diseases of aging. And it seems to be very promising. And another work that was maybe a step forward to creating more explainable machine learning biomarkers was from our group at the Gladyshev lab, combining epigenetic and gene expression data, because one of the challenges with DNA methylation is sometimes it's hard to explain, and sometimes it's changes in regions that are not functional or not associated to any genes. So to address that, we collected a large number of cohort data, including longitudinal data containing gene expression and DNA methylation. And the first interesting observation was that the same genes that are changing by age in one cohort consistently change by age in the same direction in other cohorts of diverse background, from white population to African population. And here, an example is the gene CD248 is a T-cell specific gene, and you can see it's almost the most downregulated gene by age across all these cohorts. And we expected this for DNA methylation, but we were also surprised how strong these age-dependent changes are when you have a very nice reference cohort. This is the same cohort that we built for the benchmarking, and you can see almost 95% association with age, just a single DNA methylation site in this cohort, and also being the top site associated with age across cohort. But maybe most interestingly, when you look at the epigenetic genes or sites that are most strongly associated with age, you see that most of these genes are silent and they are already repressed. And we think now the gain of methylation or the change in epigenome does not affect the function of the cells or the genes. And one explanation might be that the cells afford to gain methylation, or the cells afford to change their epigenome by age because there is no effect and there's less control over those regions, and that might be the reason these regions are so much influenced by age. And if you stratify populations based on just these epigenetic changes or these genes that are epigenetically changed by age, you don't see much differences in survival outcome. But if you combine epigenetic and transcriptomic and pick the genes that are most influenced by age on epigenetically and based on gene expression, you see this clear separation of groups that survive better or worse. And these are all functional changes, functional genes that have consequence for cells. And we found around 104 of these genes, and putting them together, we developed a simple score. And most of them capture immune related changes. Some of them captures immune cell composition, but these are less, it could be, some of them are causal, some of them are associative, purely correlative. But at the end, this could be a good marker of immune health and age-dependent changes in immune. And the other side of it is now we have a targeted panel of these 104 and plus 6 control genes, 110 genes. And we synthesized the panel in collaboration with the Twist Bio company. And now these panels cost around $30, which is 10 times lower in price. And it allows us to scale these panels across large cohorts. And another example, just to also touch base on proteomics, which is becoming a very popular addition to epigenetics, is this great resource, UK Biobank, that was profiled in collaboration in a consortium effort by some of the large pharma. And in particular, they have all whole genome data, but maybe the most interesting in the context of aging is 50,000 of these participants have been profiled using Olink proteomic assays. And for all of them, we have ICD diagnostics, lab surveys, imaging, and age-dependent outcomes in 16 years follow-up. So a nice follow-up time, nice unbiased proteomics, and this data has been used in a few very interesting research, one from Stanford, Tony Weiss-Corey lab, one from the Broad Institute, Austin, Argentina, and one from our own lab at the Gladyshev lab. And if these proteins are used to create a score, age-dependent score, you can see that many of the diseases could be modeled as accelerated aging, and some of them with very large effect size. For example, lung disease or heart failure or kidney failure. In many sense, they resemble accelerated case of aging based on the change in proteomics profile. And some of the diseases of aging, like dementia or Alzheimer, are surprisingly less strongly associated when you build these kind of signatures, accelerated aging, accelerated proteomics aging signatures. And that motivated me personally to look deeper into why that's the case. And this is an unpublished result. And we showed that one reason could be some of these proteomic expression, proteomic concentration that are causal to dementia and Alzheimer are not age dependent. Some of them are, like GDF15 is or GFAP are a good example. Both of them are a good example of age dependent proteins. GFAP here increases by age across healthy population, and in Alzheimer patients, it's accelerated. But in the case of ApoE, it's the expression, it's the protein that overall does not change by age much. And if so, it just increases a bit. But we know in Alzheimer patients, ApoE decreases in blood, and this cannot be modeled in a typical age acceleration way. And using this insight, we were able to build a more specific panel for Alzheimer that predicts the future incident of Alzheimer with really good predictive power. If you combine genomics information, polygenic risk score, demographic, and these proteomic signatures, you get close to 90% accuracy for predicting incident of Alzheimer in the next 15 years for these patients, which is clinically close to become clinically relevant. And finally, I want to mention this study that we piloted last year during our conference with the first 100 participants. And we call it simply the longevity study at Harvard. And we profile various omics for these people. And then we prospectively look at age-dependent changes over the next 5 years. We piloted last year. This year, we are planning for 300 to 500 participants to have the beginning of our longitudinal phase, depending on funding. Of course, these days, funding situations is a bit uncertain. But this might be one of the very few studies that are not funded by NIH or other federal funding. This was mostly foundations and nonprofits and collaborations with our partners at Stanford and the other group. And that's all I have for today. And I want to specifically thank Dr. Gladyshev and the Gladyshev Lab, who have been very supportive of the work, my research, and the consortium work. Maybe if you have time, I would be also happy to take a few questions.
Thank you so much. We really appreciate your time and the fascinating presentation and the unpublished data. Like, thank you so much for sharing that with our audience. We have a few questions in our Q&A box, so I know we're running low on time and you must be very busy with all that research, but if you'll allow me, I'll share a few. So the first one is coming from a person that is clearly concerned about what they can do like in the clinic regarding their biomarkers as a patient. And they ask, since aging is that highly correlated with the rate of aging is that highly correlated with how healthy they are or not, would you consider standard biomarkers that we've known for a long time in medicine, such as LDL cholesterol or CRP protein, as a biomarker of aging?
Yeah, that's a great question. And again, here I have some unique perspective that's not medically. I'm not a physician, and it might be not in agreement with a lot of other experts in our consortium. But personally, I believe that these established markers should be measured first. And for myself, I always look at these validated markers first. And these are the ones that have been benchmarked and analytically validated, and or the assays are more controlled. So personally, for my personal tracking of the health and for my family, I always look at simple markers, starting with markers of physiological well-being, weight, cholesterol, glucose, and A1C. And I trust them as a first line of evidence. And then maybe we can complement them with these new machine learning and composite markers when validated. I always say that we need to do more validation.
But do you think we can call them biomarkers of aging, or they are like they do not meet the criteria?
Yeah, so the criteria is something that as a scientific community and practitioner communities, we are defining together. But if it's a biomarker that's marked that the normal range is defined based on age. We can consider them as age-dependent biomarkers, right? If we have a normal range for younger folks that's different than a normal range for older folks, we expect to see some age-dependent changes. And that could be considered, that could be used in the context of monitoring healthy aging.
Right. Okay, great. That's fantastic. That's a good news for me too, because my blood work is overdue. So I like how you mentioned validation. That's of course like the most crucial, and I think that's one of the motivations behind the establishing of the consortium, as I've learned from some histories. So we have another question coming from a person who attended the Longevity Summit Dublin. And they say that they listened to Professor Matt Caberlain's talk, and he said that the aging clocks are fantastic, but still can be too variable on their results, given even if they measure the same things, such as DNA methylation. So, what is your thought on that? Do we have any truly validated biomarker of aging?
Yeah, no, I like that question, and Matt is one of my favorite rigorous scientists in this space. And yeah, so validation is a very broad term, and for FDA approved, clinically validated biomarker, we have around 16 different types of validation. And then the specific question, I think, was based on repeatability. And that's a measure. We might have accurate markers, but they need to be precise, meaning that if you measure it twice in a row in the same day, or same samples from the same participants, you should get very close, very similar results. And for a lot of these novel markers, these composite markers, we have not done enough repeatability. Some of these groups that I mentioned are looking into that repeatability, but we still don't have enough published evidence, and we still, some of them are more, for example, epigenetics are now more tested for repeatability, but still we don't have cross lab repeatability, cross technical replicates, biological replicates. We don't know all the variations in various scores, because every score has different number of inputs, and it's still an ongoing effort. So a lot of these markers are validated for cohort studies, and they've been validated for cross-population predictive power across cohorts, but most of them, as far as I know, are not validated for longitudinal changes in the same person robustly enough.
I see, and that seems like a very expensive and time-consuming study to conduct, right?
Yeah, and a lot of them require the follow-up time, require time and effort and cost, and we haven't seen. Probably in the next few years we'll see more of that.
Right, for sure. The power here is also very interesting, because like you can't just measure your lab members have 5 results, which would be great because they would show up, That's it's funny. There's one more question, and that's more of a clinical question than a biomarker question, but it's coming from a person who got really concerned about the 90% accuracy of Alzheimer's disease prediction from your latest model. So they ask, I am now almost, let's say if I do this test and I am now almost sure that I will get AD, what do I do?
Yeah. So I hope they're not concerned about the results, because now we see more and more of these results, and FDA just approved last month the first blood-based biomarker for Alzheimer detection. And a lot of those markers are also seen in blood many years before the onset of Alzheimer. So we think the results are pretty reliable, and but on the clinical side, unfortunately, it's a very sad story that we are seeing more and more evidence on the deterministic nature of Alzheimer. And there was a recent paper published again a month ago or very recently that showed having 2 copies of ApoE is now considered its own class of Alzheimer, because at the time of biopsy, most of them have pathology for Alzheimer. So at this point, if I think, again, genetic would be the first line of testing to see if it's heterozygous or homozygous ApoE. And if it's 2 copies of ApoE, it's based on published data. We think it's very likely that the person develops Alzheimer during their lifetime. Unfortunately, again, we don't have much strong interventions. There are some evidence on the lifestyle about sleeping and the typical healthy lifestyle factors that we know: healthy nutrition, not smoking, not excessive drinking. So we know that all of those decreases the risk, and the 90% is just the overall population percentage. So we still can change those. But one reason that I'm personally interested in Alzheimer is this unfortunate fact that we still don't have good treatment. I lost my grandma last year due to Alzheimer. So I made a commitment that I'll spend more time understanding and studying Alzheimer personally.
Right. I'm sorry to hear about your grandma, but I'm happy you managed to push that into the scientific motivation. So,
Yeah, thank you.
So this is just my thought based on what you just said. So would you agree with me if I said that the more biomarkers, let's say on Alzheimer's we have, the closer it brings us to finding quote unquote a cure or a therapy towards that, just because we would know how the algorithm assesses people on a various progression towards the disease?
Yeah, again, I'm not a neuroscientist, but as an informatician person and studying these markers in the context of dementia and neurodegenerative diseases, I think the more we can measure these diseases and pathology at the molecular level, the better we understand them, the better we stratify them, the better we treat them. And even more recently, the Alzheimer's Association and the NIA jointly defined a new framework for Alzheimer, which is based on biomarkers, the ATN framework, having amyloid, tau, and neurodegeneration. So the more these markers are measured, the more accurate even the definition of diseases are becoming.
Right, for sure. And I know we're running out of time. There's one quick silly question that I think is an excellent question that somebody asked. How do I volunteer to have my data tested to support your research? Is that even an option?
Yeah, so we are going to collect samples again during our conference. So if they are near Boston or they can join the conference or even they don't need to attend the conference at the same time of the conference in the same venue, we are going to do sample collection, and we are planning to profile all the omics. And last year, we measured some functional tests. This year, we are planning to also measure cognitive tests. And we also have a very comprehensive surveys of all the health status and medication and everything else, so we can connect them.
Okay, well, that's the call to action then. Biomarkers of Aging Consortium. Is it November, September?
And this year is in October 20th at Harvard Medical.
October 20, beautiful. Thank you so much for your time, Mahdi. I really appreciate it. This was such a scientifically dense presentation. I will definitely go back to it and make some notes for myself. Thank you for your time. Thank you for answering all the questions from our participants. It's been a true pleasure and honor to have you in our hub. And please come again if you want, please.
Thank you very much. Thank you, and thank you, Evelyne.
Thank you, Dr. Moqri. Thank you. Really great pleasure and great wealth of wisdom. See you at ARDD.
See you soon.
Have
a good day.
Thank you. And for everybody who's still in our webinar, please follow Dr. Moqri's research on Google Scholar, and please welcome to our Longevity Education Hub at longevity.degree to take the courses that are free and fully volunteer-led, but also scientifically validated by the physicians. So as much as AI is great, we often rely on physicians to test our content for scientific and medical reliability. Thank you so much for your time, and we will see you on the other side of the internet. Thank you. Goodbye, everybody.
Yeah, bye-bye.